Carnegie scientists explore the outermost reaches of space, the mysterious depths of the Earth, and the origins and mechanisms of life. Our brilliant and dynamic teams of scientists transform the landscape of discovery with their unbridled passion, insatiable curiosity, and never-ending quest for knowledge. With limited teaching, administrative, and grant pressures, Carnegie's scientists concentrate on research and are empowered to go their own way. Our scientists are chosen for unique skills and boldness. Their amazing discoveries, spanning more than a century, demonstrate the power of freedom. Carnegie astronomers, astrophysicists, and planetary scientists are committed to finding answers about our universe. From the expansion of the universe, discovered by Carnegie's Edwin Hubble, to the chemistry of stars and the diversity of exoplanets, Carnegie's distinguished investigators probe distant reaches of space and time. From curiosity comes discovery. Geophysicists, geochemists, and ecologists use cutting-edge instrumentation in our laboratories and venture out to nature's laboratories, stunning rainforests, ancient rock formations, flowing lava lakes, and beckoning coral reefs. They explore Earth history and dynamics with passion and determination. Unprecedented independence from short-term pressures, close collaboration, and interdisciplinary approaches set Carnegie scientists apart. Taking risks and using new approaches, their findings have critical implications for understanding Earth's properties and guiding policy. Carnegie scientists make outsized contributions to unlocking the secrets of life and laying the foundation for improving it. Carnegie's Nobel Prize-winning Barbara McClintock made discoveries about chromosomes that completely changed biology and medicine. With latitude to undertake long-term, higher-risk projects, our researchers investigate the molecular basis of life, from computation and genomics to advanced microscopy and engineering, to remote sensing tools and forest canopy fieldwork. We construct a clearer picture of plant, animal, and microbial life, diverse ecosystems, and changes driven by human activities. Carnegie investigators are intrepid adventurers who pursue their goals even as they mentor and train the next generation of scientists. They are free to question, to wonder, to creatively pursue ideas from the vast universe to the subatomic world. Our scientists redefine the pursuit of what's possible for us, our planet, our universe. We explore the past to understand the present and inform the future.
Welcome, everyone. Welcome, everyone. You can hear me, right? My name is Yushen Zheng. I'm the director of Carnegie's Embryology department. This is our first public lecture, and we are starting this because we have come to recognize the public really should know what we're doing in our daily work of doing science. And there's also a very strong interest to really want to understand how we scientists conduct science and how it's not. Oh, okay, sorry. Okay, so you have seen a brief video. What I'm going to do is to give you a very brief introduction to this department of embryology. Carnegie has 3 biology departments with this embryology department on Hopkins campus. We've been affiliated with Hopkins campus since 1914. Our first home was actually at the medical campus at Johns Hopkins. In the mid-50s, we moved to the Homewood campus. This is the building that's just right up the street of San Martin Drive. We've been in that building for several decades. And the institution built a new building here, just in this current building. We moved into this building since 2005. Our department has a total of 8 faculty members, staff members, what we call it. It's equivalent to the faculty position or professor position in universities. we also have between 2 to 4 staff associates. The staff associates are the positions we offer to bright young scientists who have unique ideas and want to pursue their independent research program. And here are some notable recognitions of our department. We currently have 2 Lasker awardees, 1 Gruber Prize. Since the 70s, 10 staff members have been inducted to the National Academy of Sciences. And since the 80s, the department has hosted 7 Howard Hughes medical investigators, and we've gotten 1 Nobel laureate, and that was awarded in 2004. I'm going to come back to this just in a minute. The department is well known for its ability and for its history of developing model organisms for in-depth biology study. And here I've listed 6 of them: Drosophila, nematode, mouse, baker's yeast, and zebrafish, as well as planarian. For biology, for what we know about biology, really a lot of the knowledge really came from the study of these unique model organisms. In order to study them, we have to develop tools, and that's what the department is very well known for. And more recently, some colleagues of ours in the department have begun to develop tools to try to understand the coral reef organisms in hoping to really better understand how coral reef can handle the ever-increasing environmental challenges such as global warming. So it is actually through the use of this nematode, Dr. Andy Fire, who was our former colleague, along with his collaborator, Dr. Craig Mello, who have together discovered that small RNAs, in fact, play a very important role in regulating gene expression. And this discovery is really very important in the understanding of biology in general. It also has created tools to allow biologists to be able to manipulate gene expression. For this discovery, a Nobel Prize was given to both Andy Fire and Craig Mello. So now this is my brief introduction. I'm going to hand the podium to my colleague, Dr. Marnie Halpern, who is the neuroscientist who uses zebrafish to study how the brains work. She'll introduce our speakers, and she will also moderate today's discussion. Marnie.
Thank you. Okay, well, I'm going to turn the lights off if that's okay. I want you to see me. Well, first of all, I'd like to reiterate Yixin's welcome and welcome you to our new neighborhood speakers series. Tonight, we're hearing about a topic that I expect will be of interest to everyone in the audience because we all have to go through it, the process of aging. Whoops, where am I? Oh, here. As a famous movie star once said, getting old is not for sissies. And there's a lot of truth to that. In fact, we've long been fascinated by ways to overcome the process of aging by searching for example, the fountain of youth. Whether it's just magical waters or reinvigorated stem cells, we wouldn't mind a positive outcome, a miraculous reversal of the aging process, as shown in this old postcard from Florida. Of course, this might be a lot to ask for, a reversal. But if we could just come up with some better ways to fix old or damaged cells by repairing or replacing them with new ones, we'd be quite happy. But the question is, does such a healing factor only apply to comic book characters like Wolverine? As you can see, he's regenerating his inside there. Or the Incredible Hulk, who has the complete regeneration going on. Or will modern medicine be capitalizing more on healing factors in the near future, using what we've learned from our research on those animals that do have the remarkable ability to regenerate? This evening, we're fortunate to have 2 distinguished scientists talk to us about their own research related to aging and regeneration. They will each give a short presentation, followed by what I hope will be a lively discussion with questions from you, the audience. And we'll have a microphone. You'll be able to come up and ask any question you want, but maybe not about astronomy. We'll stick to aging and regeneration. Our first speaker, Dr. Alejandro Sánchez Alvarado, is currently an investigator of the Howard Hughes Medical Institute, leading a research group at the Stowers Institute for Medical Research in Kansas. Alejandro, a native of Venezuela, obtained his Bachelor of Science degree at Vanderbilt University and his PhD at the University of Cincinnati College of Medicine. Alejandro is well known to us here at the Carnegie because he was briefly a postdoctoral associate before starting his own research group as one of those brilliant staff associates you heard about in this department. It was during this time and later, after he moved to the University of Utah School of Medicine, that Alejandro began to tackle the problem of regeneration, bringing new cutting-edge techniques to an existing animal model, and as you'll hear about tonight, exploring new animal models as well. Alejandro is the recipient of numerous grants, honors, and awards. There are too many to mention tonight. But notably, he was just this year elected as a member of the National Academy of Sciences. So congratulations, Alejandro. It's a real pleasure to have you back at the Carnegie.
Thank you very much, Marnie. And thank you very much for the invitation to be here tonight. And thank you very much for all of you to come and listen to what we have to say. I understand this is going to be in YouTube at some point, and it's my mom's birthday today, so if you could just allow me for just a second. Feliz cumpleaños, Mama. Because she's going to be watching this. Okay? All right. So what I would like to talk to you about is something that you may not really ask yourself every morning, but it's something that I've been asking myself for quite some time. And so have you ever wondered why you can recognize yourself in the mirror? Every morning you go to the bathroom to brush your teeth or whatever. I find that to be truly an interesting question because we are composed of millions of cells, and somehow from day to day we can really recognize each other. I can recognize Marty, I haven't seen in a long time. Marty can recognize me. And of course our family can recognize us on any given time. And this is something that we really don't understand in great detail at all. And in fact, parents are fascinated by this. So I'm going to show you this condensed version of a time-lapse movie that a dad in the Netherlands did of one of his daughters. Her name is Lotte, where essentially he just took a picture of her every day for like 16 years. And I want to stop it on year 1. You can see the years are going to be progressing. And what is remarkable about this is that you recognize her as being that little girl. So what I find astonishing is that you start as a beautiful baby, you go through these tremendous changes that we all know are upon us. You know, they are growth, learning how to speak, learning how to communicate emotions, adolescence, all these changes in your physiology. As you're growing up and mature, year 11, you can see her here, and you can see clearly that this person is changing on a day-by-day basis. And the remarkable thing is that as this person gets older and becomes a person of her own, you know, to be brave enough to have a picture like this shown right here. I mean, I'm sure this was, and then end up like this. This is beautiful. This is a process we all take for granted. We think this is fantastic when we do it, and when we see our children do this. But the remarkable thing is that we do not know with certainty how this process unfolds. We essentially have no idea. Sorry, my microphone was off. Okay, now it's on. Wonderful. So we essentially have no idea why does it take this long, for example. Just a basic fundamental question. Why is it that it takes this long? Why does it have to take 15, 16 years? And why is it that that biology will continue for as long as it may to say, for example, 80 years of age, or however long we hope and aspire to live? That we don't understand. It remains a mystery. There's a lot of changes that are taking place in your body, and all of those changes have to be occurring in such a way that you can actually recognize yourself the next day that you see yourself in the mirror. Okay, so I'm going to illustrate what I'm trying to convey to you by essentially telling you that I can pick any multicellular organism on the face of this planet. And what we're going to talk about, this process of temporal transformation of your tissues, is so fundamental that I can go to the most inhospitable place in the planet, find a life form there, and actually show you what I mean by this. So we're going to take a trip, a virtual trip, to the Atacama Desert on the coast of Chile, in the high plains of the Andes. It's probably one of the driest places on the planet. They see water every decade or so. They get most of the moisture from the ocean, but it truly is a high altitude desert. There's not enough time for water to condense, and yet in this inhospitable and hostile environment, you find life forms. So I want to show you one example of such a life form. This is a plant that botanists call Azorella compacta. And the reason why I will show you this plant is because even in this inhospitable place, it's actually doing the things that we're interested in trying to understand. So I'm just going to draw a simple timeline underneath the plant, and I'm going to start the history of this plant in 1988, which is one of the first time that an extrasolar planet was described in the literature. Okay? This plant was growing happily in the Atacama Desert in 1988. Well, in 2018, that plant's still there. In 1945, when the first atomic explosion was detonated, that plant was growing just fine on the Atacama Desert. That already makes the plant close to 100 years old. When Darwin was first drafting his theory of evolution, that plant was growing, in its own remarkable way on these plains of the Atacama Desert, making the plant already over 100, 150 years old. When Galileo aimed his telescope at the sky for the first time, okay, that plant was growing in the Atacama Desert. That makes the plant already 400 years old. You can go even further, okay, all the way down to like the Black Plague, when like almost a quarter of Europe was being decimated by bacteria and by a horrible plague. This plant didn't care. It didn't care that Europe was falling apart. The Western canon was on the edge of disappearing. It was still doing its thing, growing as a plant. In fact, I can go all the way back to the writing of Summa Theologica by Thomas Aquinas, whom I actually blame for everything else from there forward. But in 1273, this plant was probably also growing and living just happily on the Atacama Desert. I'm going to stop the history lesson here by just telling you that. Just think about this. This plant was alive throughout the rise and fall of the Roman Empire. Right there in the backwoods of the Mediterranean, when the world was small and people knew nothing about what was going around their environment, the planet, the universe, etc., etc. This plant was doing its thing in the Atacama Desert. This plant has been estimated to be approximately 3,000 years old. And this is not uncommon to plants. Even sequoias, some sequoias on the western United States on the Pacific coast have been estimated to be 2,000, 3,000 years old. So how is it that these organisms can actually do what they do and live for so long? These are mysteries that we really don't understand, and we have hypotheses, plenty of them, but we need to test them, obviously. So one of the things that becomes apparent when you inspect this plant closely, and you look at how it's being formed, is actually composed of these rosette units of individual little plantains, okay? And inside of each of these plants, at the very apex of each of these, there's a structure known as an apical meristem that contains a collection of cells that are responsible for producing all of the cell types in this plant. Those are plant stem cells. Stem cells are an old evolutionary invention that's been around since before the split between the plant kingdom and the animal kingdom, and they clearly play a fundamental role in the longevity and the maintenance of organisms all over the planet. So if you now reduce this question to maybe the simplest thing, let's think about a simple organism like bacteria. In the case of bacteria, in order for it to survive for a long time, it essentially just self-renew, essentially just divides into two. And you can see that in this stock photography of bacteria undergoing cell division. And you can see essentially how within a very short period of time, a single or a small group of bacteria can actually produce thousands, if not more. Every 20 minutes or so, this bacteria will be dividing. And that's the way unicellular organisms solve the problem of self-renewal. They just copy themselves and make more of themselves because they're just simple single organisms. That is not true when you look at complex multicellular organisms. You start like a single cell, just like a bacteria. The cell begins to self-renew, just like a bacteria would do. But as this process unfolds, you begin to see that some of these cells begin to change shapes and begin to distinguish themselves from each other. They're not all homogeneously structured. And what is more remarkable is that as this process unfolds, the final product does not look anything like the early product. So this embryo is an embryo of this organism. This is a ctenophore. And you can see there's no indication that these animals have this rave party going on in embryogenesis. It's happening actually in the adult. And then this is something that is truly, truly astonishing. And this is where what we do becomes really important, because this is what development is. You go from a single cell, you go through this process of development, which is some really interesting prism that actually deconvolves entirely all of that mystery in a single cell to produce a completely well-formed organism time and time again. And this I find absolutely astonishing. And the fact that these animals can live for a long time with almost no protection in the middle of the ocean and perpetuate themselves is really indicative of the remarkable power of this process that is self-renewal, tissue homeostasis, and tissue maintenance. So let's just step back and think about us. Let's go back to Lore, okay? So, you know, she will go through adolescence, become an adult, go to college, get a job, get married, et cetera, et cetera. And what's happening when you become an adult, like most of us here in the room, we know, and it has been estimated that in a single day, each and every one of us will lose anywhere between 50 to 70 billion cells. These are American billion, these are not metric billions, okay? So it's just 50 millions, okay? 50,000 million. So anyways, if you do the math, okay, the number of cells that you lose in a cell, in a year, sorry, will correspond to a mass that's equivalent to the entire body weight. Now just think about that for 1 minute. If I lost this many cells today, 50 to 70 billion cells, I woke up this morning and I looked at myself and I recognized myself in the mirror again, how did that happen? Why is it that we are able to maintain this really apparently very stable outward appearance when it is belied by constant change? That is one of the things that we really don't understand and we would like to understand in much greater detail. So how do we know that this happens? Well, people have been very clever. And so a group of scientists in Stockholm at the Karolinska Institute did the following experiment. They reasoned that if they used atomic explosions that we humans have been detonated for a long time, since it's going to be carbon-14 in the atmosphere, it's going to be absorbed by individuals, and that's going to be part of their DNA, and then you could measure how old those molecules are in any given individual. And you can see that if you measure carbon-14 all the way from the beginning of the recorded history to the present time, there's a huge spike somewhere in the middle of the century. That's since 1945. If you now do the following, which is that let's go look at a tree trunk, and each of those rings corresponds to a year. Let's find a tree that was born at or about 1945 or 1950, and then remove a sample of wood, and then measure the carbon-14 in each of these rings. And what they find is really what you would expect is that beginning 1950, it rises all the way to the very top, and then it drops. This is the signing of the Seoul Treaty, when no longer atmospheric explosions were allowed. You have to be subterranean. So you actually see a real significant drop of carbon-14 in the atmosphere. So that told them that we have a clock that we can use, a marker, a fiduciary marker, if you will, that allows you to find out how quickly these carbon molecules may be turning over in an individual. So they did the following. They went and they looked at humans that were born right around 1950s, 1960. I believe one of them in 1964. And then you essentially ask, you know, how much carbon-14 do they have in their tissues? And they did this in biopsies. They also did in individuals just died from disease or accidents or natural causes. And eventually what they find essentially is that in fact some of these tissues are turning over significantly and others are not. So I'm going to use Google Man. This is a guy that I hope he knows he's in Google, but I'm going to use him as an example of what I mean by this turnover. So the cells of our body appear, and it's pretty clear now, that they turn over at very different rates, okay? So if you look at the frontal lobe of some of these patients and you find that they were born in 1964, they still have significant amounts of carbon-14. Suggesting those tissues are not really turning over very, very quickly. If you continue to look at other tissues like the heart, that's also going to be years. You still find carbon-14 in some of the cells or tissues of the heart. The liver is 300 to 500 days. We know that from different measurements in various other fields. Of course, muscle, depending on whether you're sedentary or active, that also turns over rather readily. And this is something that Fan is going to talk to you about very soon. And then, of course, our red blood cells, every 120 days, every 3 months, are turning over at a remarkable rate. The bottom line is that, and of course, my favorite, the gut. The lining of the large intestine is the surface area is equivalent to the surface of a tennis court. That gets resurfaced every 5 days. Now, you just think about this. Why aren't we having more problems, okay? I mean, if every 5 days I'm repaving my stomach and my gut, I mean, it's got to go wrong at some point. And I'm right now 54 and it hasn't gone wrong. So it's really astonishing that every 5 days this has happened. And maybe when you're a child, it's even faster. So anyways, if you take the collective of all of these cells, the average age of a cell in an adult can be anywhere between 7 to 10 years. So I may be 54, but I have a cell that was just born. So how old is that cell? I mean, to what extent do we really want to think about biological time versus astronomical time? And are these two things really related to each other or not? And we really don't know. We suspect that they may not, or they may be in some circumstances, but we really cannot say with authority whether or not these two things are in one way related to another. So when Lewis Carroll wrote this in Alice in Adventures in Wonderland, even though it was supposed to be funny, turns out he was pretty spot on. At least I know who I was when I got up this morning, but I think I must have changed several times since then. Yes, you have. Believe it or not, you have. Okay, so these are really fundamental problems in biology. So how can we best study and solve these problems? So let's put ourselves in perspective first, because it is important for us to realize that the universe was not made for us. Right? We like to think that. We really believe that everything gravitates around humans, but that's not really the way nature and the world has actually worked. And this is a tree of life that contains about 6,000 different species. And essentially the message here is, from sequencing so many genomes, is that all of these different animals and organisms and plants and yeast and so forth appear to share a common origin. It didn't have to work out that way, but that appears to be what the data is telling us. So you can see here the collection of plants, protists, bacteria, archaea. You can see fungi, and somebody loves fungi. And then, of course, animals up here. And now you ask, okay, we're such a great species. I mean, we should really occupy a prominent spot. We don't. We're buried right there in between worms and other critters and so forth and so on. So it is just our own arrogance that makes us believe that, we should really occupy a very important spot. It's just our history. But we will really occupy a fairly unassuming location in this tree. So if that is true, if we share all this common ancestry and we share all of these common mechanisms, it should be possible for us to try to investigate and dissect these processes in systems where this biology is most readily accessible. And so I'm going to show you an example of the animal that we developed or began to develop as a system right here at the Carnegie Institution a few years ago. I hate to say it's almost 20 years ago, but that's how time flies. But what's going on here is, and I heard you squirm already, is that we took a worm and we used a razor blade, which evolution I'm sure did not use, and we cut it into 3 fragments, okay? And what's going to happen to each and every one of these fragments is really, really astonishing. This animal, under normal conditions, by our own assumptions, would be dead, but they're not dead. They'll actually regenerate complete animals. I'm going to give you an extreme example. I cut this animal into 18 different fragments, and each and every one of those fragments, in the span of essentially just 2 weeks, produced 18 complete animals, okay? That is the equivalent of me cutting my little finger and watch my little finger regenerate me. That's what these guys are doing. And so to study this process of aging and restoration of tissues, why not go to animals that regenerate? Because it truly is an exaggeration of this process, and it is compressed in time. So we should be able to resolve or address these questions at a more mechanistic level. So one of the things that we were able to develop in this system by adopting methodologies that were developed by Andy Fire at the Carnegie Institution was to try to perturb the functions of these genes to see what activity they may or may not have in the biology of regeneration that these animals display in such great abundance. So here's an animal that was amputated in 3 different fragments. And what you can see here in this movie is that each of those fragments, tail, and trunk, essentially just go on to regenerate complete animals. They do it like nothing, okay? Like you and I are breathing or chewing gum and walking at the same time. They just do it incredibly well. And so what we figure is that if we could perturb normal biological processes that are responsible for these remarkable regenerative capacities, we should be able to learn something remarkable about this. I'm going to talk to you about this molecule, which is known as beta-catenin, which is involved also in tumors in humans, okay? And this molecule has been crystallized. We know a lot about it in many systems, including humans. But what we can do in these animals is that we can get rid of this molecule, amputate the animal, and then ask, what is the absence of this molecule going to have in the ability of these animals to regenerate? And to our surprise, what we were able to do was to generate animals that now have heads at both ends. So elimination of a single molecule was enough to confuse the cells and not restore the missing body parts like the tail, but turn the tail into a head. I almost fell off my chair the first time that I saw this phenotype. It still hurts. So we can do the opposite experiment, which is, what if we now accumulate this molecule inside of the cell? Let's not get rid of it. Let's just accumulate it and then see what would this excess have as a consequence on the ability of these animals to regenerate. And at this point, I should say I fell off the floor, because the animals, instead of regenerating heads at both ends, they regenerate tails at both ends. So essentially what we find is that we have a single molecule that can actually throw a switch of sorts that tells the cells in this animal to either make a tail or make a head. And that's the power of being able to investigate these types of biological processes in animals that truly exaggerate the biological problem that you want to investigate. Now, things got a little bit more interesting when we decided, okay, well, let's get rid of the molecule, but this time we're not going to cut the animal. What would we get? Because this would address as to whether or not this particular molecule has any role in just normal tissue turnover. And when we do that, and we get rid of the molecule, this is what we get. We get animals now beginning to grow heads everywhere. At that point, I fell off the building, okay? Because I was not expecting this. I mean, we were supposed to have a situation where this particular molecule had to be activated by some stimulus. That stimulus may be being amputation. But what this tells us is that this pathway is always on, that this molecule is working all the time to maintain the form and function of this animal so it can be recognized as a planaria at any given time. So that gives you an idea of how interesting and remarkable it is to be able to have a system where you can really mechanistically and at high granularity begin to dissect this really fascinating biological problem. So the reason why these animals can do what they can do is because in their body plan, they actually possess a large collection of stem cells. And unlike the stem cells that we have in our body as adults, these stem cells can actually give rise to everything. So you're looking right now at a planarian stem cell. If you inject this cell into a single animal, that single cell can produce every tissue type, every single one that's present in the animal. Human stem cells, as adults, they have much more limited potential. So how is it that these animals were able to retain their ability to access the entire genomic output in a controlled fashion to maintain form and function of these animals? And this is the beautiful thing about this deep conservation of function that I've been trying to impinge upon you by showing you that tree of life, is that they have genes. These genes are in DNA. We can clone all this. We can ask all these questions, and we can actually get to understanding how these genes actually do what they do. So I think these are really fantastic times for doing these kinds of research. And I think what I would like to impinge upon you is the following, is that we have just barely scratched the surface. We have not really gone to find out what is truly possible in biology out there. And I'm just going to give you an example from a very recent set of discoveries. So here is the first extrasolar planet. It's in Gamma Cepheus, which is shown right there. That was published by Campbell, Walker, and Yang, and a lot of people didn't believe this was true. Now we know that there's a lot of extrasolar planets out there. But what I find really astonishing is that that same year, this other thing was discovered, Prochlorococcus marinus, okay? You can see this from the space station. This is probably one of the most abundant organisms on Earth, and you should be shocked that it was only discovered in 1988. We are not looking. This is the problem. We are not looking. We assume that we know what we need to know, and armed with those tools, we can go in and explore. But the bottom line is that we are not really looking in any meticulous and systematic way to try to understand what is already possible in the laboratory of nature. And so this was discovered by Peggy Chisholm, and this is essentially it's a green-blue algae, and it's incredibly, incredibly abundant. So here's what I would like to end with, is that we have made incredible strides in understanding our own biology by focusing primarily on 7 single model organisms, okay? So we have humans, chicks, frogs, fish, mice, C. elegans, and flies. And we're not done with this yet. There's way more biology that is probably going to be coaxed out of these organisms that will help us understand our own. But if you now do the math and you assume that there's about 9 million or so species in the planet, these animals represent only 9 times 10 to the minus 5% of all known animal species. Now I ask you a statistical question. What are the chances that everything that we need to know about biology will be present in 9 times 5 to the 9 to the minus 5%? It's essentially 0. That's exactly right. And so that is a conundrum, because we've made a lot of progress, but yet we know that we have a lot of things that we don't even know exist, but they're already there, and nature has been doing a great deal of that. In fact, new species are discovered every year. Many of these completely new to science. We don't know much about their biology. We don't know about how the chisel of evolution may have produced very specific functions for well-known and unknown molecules, and how they come together to produce form and function and give this immense diversity of life that we immediately recognize as being beautiful, which is another thing that is also hard to understand. So I would like to conclude by just giving you a quote, because I think that our specialization has begun to, at best, inhibit our ability to ask different questions, and at worst, really hurt our ability to progress our scientific understanding. So this is from a science fiction writer called Robert Heinlein, and he says, or he wrote, that a human being should be able to change a diaper, plan an invasion, butcher a hog, con a ship, design a building, write a sonnet, balance accounts, build a wall, set a bone, comfort the dying, take orders, give orders, cooperate, act alone, solve equations, analyze a new problem, pitch manure, why not, program a computer, cook a tasty meal, fight efficiently, and die gallantly. And then the concluding sentence is that specialization is for insects, which we clearly are not. And so with that, I conclude, and I look forward to your questions after Fan's presentation. Thank you.
Okay, thank you, Alejandro. So we're going to move now from worms to mammals. Our next speaker, my colleague, Dr. Chen-Ming Fan, is a staff member in this department and an adjunct professor in the Department of Biology at Johns Hopkins University. He's originally from Taiwan. He received his Bachelor of Science degree from the National Taiwan University and his PhD from Harvard. He was a postdoctoral fellow of the University of California in San Francisco before joining the Carnegie. Fan has received a number of awards for his research and significant support from the National Institutes of Health for his innovative studies on muscle cells, which you're going to learn about now.
Thank you, Marnie, for your very nice introduction, and thanks everyone for coming in this evening. Also, I would like to thank all the organizers, including Marian and Milan, doing such a wonderful job for making this to happen. So in contrast to what Alejandro just told you with all the beautiful creatures, today I will only focus on one single tissue, that is the skeletal muscle, that we use every day, including I am standing up and you are sitting down. So I think that, so some of you may have went to the Body World display about 10 years ago coming to Baltimore. And I want to point out that this is all the, I think it's cadavers, actually the skin was removed. And I hope you appreciate either for artistic expression, dancing, or really, really difficult gymnastic maneuvers, skeletal muscle is absolutely essential for you to do those. And this is just a promo from Baltimore Sun. So since we're in Baltimore, for Mr. Anatomy with a body paint. But what I really want to end with some kind of weird handstand, what I really want you to appreciate is that even though the red part is your muscle that can move, muscle couldn't move our body without contacting or directly connecting to the white part, that's the tendon and your bone. So Carnegie scientists have been very interested in muscle research and can be dated into at 1963. Erwin Königsberg is among the first to actually isolate chicken muscle cells and culture in a dish. And this is a single cell that, just like what Alejandro said, they can divide to be many cells. Eventually, many cells fuse together and form these fibers called the muscle fibers. And even more days later, they create these sort of swirling patterns of many muscle fibers. I hope you can see, I don't think you can, This is a high magnification of one of these fibers. I hope you can see all those white lines. And I would ask you to recall from your high school education, those are called the sarcomeres. They can expand or contract, and that's how your muscle can move. Of course, without tendon and bone, they can't. So this approach since 1963 has been continuously developed, and in the near future, you should be able to taste the burgers and chicken nuggets completely made from the lab. I think at this point it's still too pricey for me, but it's predicted in 2021 it will be about $11 per lab-made burger. So the other thing Carnegie really having, especially this department, has a tremendous contribution to human development, is to be able to stage the human embryos for many reasons. Like Alejandro said, we have model organisms to simulate human development. Over the years, it has transitioned to most of the time we use the mouse as a model. And of course, that's because there are tremendous similarities and there are also differences. So over the years, we have used the mouse as a model for many years, and there are many, many techniques that can be developed to study them. So this just shows sort of self-promotion to show the data from my own slide. This is corresponding to this stage of these embryos. So here we can use a technique to mark the muscle in blue. And here, these are the muscles for your vertebra. And eventually you will see the ribs muscle coming up, and that's the cheek muscle. And that's the limb muscle right around here. And you can see continuous development over time. So other than we can mark the muscle, mouse has been a great model system. It's one thing is we can make mutation in any gene and any time we want. And then look at the phenotype. I mean, look at what the consequence is. So again, we have to mention one of the Carnegie staff associate, Se-Jin Lee, who actually started this work here with Donald Brown, and then move on to Johns Hopkins Medical School and discover one of the most important gene, I would have to say most important gene in muscle. And what he found is when he mutate the myostatin gene, so this is a typical mouse, and this is a mighty mouse, which is a myostatin mutant. And this is just look at the muscle here. This is about 4 times bigger for most of the muscle group. And this is really astonishing in that later on he find actually the mutations exist in nature. This is a Belgian blue bull, and you can see how much muscle it is. And this is a poor man's racing dog, a whippet. This is a normal one, and this is a mutant. And of course, later on, he actually collaborated with many people to discover there's one single human actually carry the mutation in the myostatin gene as well. And this is the newborn. They already have a lot of muscles. So I think this patient at this point, this person at this point is about 19 years old. So of course, you don't have to be a mutant to have lots of muscle. You can always hit the gym and gain muscle weight, not me. It's not going to work for me. So this is, but you can do that. But no matter what, unlike things can live through thousands of years, we don't. Even Arnold have to age, and we all age. And this is one of the skins through the thigh. This is the bone, and this is the skin, and the gray area, the dark gray area is your muscle. And in the aged individuals, I hope you can appreciate the muscle are much smaller. And in between the muscle are a lot of white lines, and that's your fatty tissue as well as fibrosis. And that phenomenon is called sarcopenia. So we do now recognize that the reason we we lose muscle as well as ability to regenerate as you grow older, every time you hurt yourself, it takes a lot longer to regenerate. They are all muscle stem cell issues. So my lab actually identified the muscle stem cells in the mouse, and we are very interested in studying how does the muscle regenerate, and why does the muscle regenerate so poorly in the aged mouse. So I'm going to show you a few videos to show how we videotape how muscle stem cell regenerate inside of a living mouse. So this is our high-end microscope suite, I think. And these are all the computers, and this is the scope. And the mouse is actually here underneath the thermal blanket, because doing the imaging, we have to keep them the same temperature as the body temperature. And we image right around here, and this is put into sleep. So in the next video I'm going to show you is muscle stem cell. We actually can mark them to express green fluorescent protein, so they will be green. So this is a video for condensed from 8 hours. So I hope you appreciate each of these green cells are the stem cells, and these red tubes are the muscle fibers. And they really do absolutely nothing. They're quite boring. This is just a high magnification of this image, and this is the muscle stem cell. You see the really long arms. And these lines are, again, the sarcomeres I told you before in the dish as well. However, if you damage this muscle, all these will be gone, except the stem cells. So this is at 2 days after we injure them. I hope you can appreciate the cell actually divide and they move around. And what I also hope you appreciate, they actually follow these red cables. And then wait a minute, I will tell you what they are. A day later, I already told you what they are. So a day later, you will see a lot more stem cells. They actually proliferate, gets more. And 1 day later, they actually fuse to become these long muscle fibers. So it turned out to be these red lines. I hope you can appreciate this fiber is flanked by these red lines. And it turned out to be these are the muscle leftovers. So you damage the muscle, there's something left over from the outside of the cell, and we call them the ghost fibers. And the next slide, I'm going to summarize all the data we have from that study. So what we think is that, and in this slide, I actually depict them in blue. So we think what happened is that when you damage these two muscle fibers, muscle stem cell will divide, and they will move around to fill the tube, and they will fuse together and to form new fibers. And they do everything inside of the ghost fiber. So this becomes very important to consider the concept called proportional regeneration. And I think all of you guys know, that is when you damage two fibers, how does the muscle know only generate two? They don't regenerate to 100, they don't not regenerate. So we think if once you have the leftover as the blueprint, you always go back to exactly the same number. This is also have a strong prediction that is if you remove the muscle together with the ghost fiber, then the muscle stem cell couldn't do anything. And this is depicted here. When you remove the muscle in this part, you can see an indent of this soldier. So when this soldier actually have the bomb blasted off the muscle, because the whole thing is gone, they can actually not regenerate. And that actually medical term is called volumetric loss. So that means when you lose the whole thing, you can't really regenerate. So how does the muscle stem cell cling onto the ghost fiber so they don't lose it? So one way to think about it is they have proteins on their surface to grab onto the ghost fiber. So we have studied one molecule called integrins. Those are these red or colors. So this is a blown up high-level structure of the integrins. So integrins come into two flavors. Either the extended form go up to grab the ghost fiber, that's called the active form, or the one bent it over and they don't contact the ghost fiber, and they're called the inactive form. It's the switching between the active and inactive form allows the cell to move along the ghost fiber. So what we were able to show, that is we remove the ghost fiber, we remove the integrin on the muscle stem cell, there's virtually no regeneration. While the normal regeneration, these are cross sections, so you can see each of the regenerative fiber are these squares outlined here, one in yellow. So integrin is absolutely essential. So now we introduce the idea with there are specialized muscle stem cells, there are ghost fibers, and there are integrins. They are all important for muscle regeneration. So which one is the one that become defective during aging. That's why we couldn't regenerate. The answer, as all biology goes, is all of them. So in fact, in the aged muscle, there are about a quarter of muscle stem cell left. In fact, there are a lot less ghost fiber materials, about 40-fold lower. But integrin is a little bit weird. We've done the sequencing, we've done all the comparison. Integrin is actually not reduced. So I'm going to tell you a little bit weird thing about integrin. So we can look at the integrin. So I remember I told you there are active form as well as inactive form. So here's again, green cells are the stem cell. In the stem cell, we probe for active integrin. And I hope you can appreciate it is aligned here. And the white line are the ghost fibers. Just to show you the active integrin is actually on the surface contacting the ghost fiber. When you look at the aged ones, there are some, but it's just not really aligned to grab to the ghost fiber. So this perhaps explains that when you injure the young one, they regenerate really well. Here's the injured area, and these are regenerated fibers. In the aged one, they don't regenerate well. There are some very, very tiny fibers. So aged people do regenerate. It's just much less. So however, to our surprise, when we probe for the inactive integrin in the aged mice, there's actually plenty of them. So it looks like because there's not enough ghost fiber, they actually, most of the time, they couldn't grab on anything, so they become inactive. So there is one prediction from that, is that if we can somehow make it extended to bind to the whatever ghost fiber they can get, we should be able to improve the muscle regeneration. And it happens to be there is a reagent. It's unimportant what it is, other than we call it TS216. It can bind to the bent integrin and make it extended. And we are very happy to see that when we apply this to the aged muscle, they actually regenerate just as well as the young one. So as I said, mouse is used as a model for human. So I want to bring you back to what is the relevance when we study the mouse model for human health. And I think I'm going to give you two examples that I think you commonly hear. One is cancer cachexia. That is people who have late stage of cancer. The cancer load will induce muscle as well as fat tissue loss. In fact, which come back to the myostatin. So regardless what the title says, we scientists like to use jargons, but the reality is this activin 2B receptor antagonism is basically to inhibit myostatin. So in mouse, this has been shown very, very effective to extend the mouse with tumor with the length of the lifetime as well as increase their muscle mass. So the other one commonly you've seen are muscular dystrophies. Those are genetic mutations that cause muscle weakness, and often the kids have to be in wheelchair at a certain time. So in our study, using the integrin to enhance integrin and enhance aged muscle regeneration, we show the same principle apply. We can also inject the TS216 to activate beta integrin, and we can also improve muscular dystrophy in mouse. And we have not gone through a clinical trial, but this one actually is in the clinical trial phase by Amgen. With that, I would like to thank all the people in my lab. Without them, it's not possible to have anything done. Of course, all the wonderful collaborators I have in the past, and lastly, the funding both from Carnegie as well as from NIH. And last but not the least, I would like to thank Ed and Jeremy for not only IT support, but also artistic support, because they helped me a lot with the slides.
Go to the next
and thank
slide.
you.
Okay. Okay, so now it's your turn. We're just going to take a couple of minutes to get our speakers up here at the front and get the microphone set up. So I want all of you to start thinking of, I know I have a good question for them, so I'll start it off. But
why am I not surprised?
we're going to move the microphone to the middle and people can come up and ask questions.
Oh.
So I'm going to start. No, I was just reading, based on what you showed, Alejandro, about this carbon dating, that there are sharks, or is it sharks or whales in Greenland? No, I was just reading, it was in Science a few years ago, that they dated them to be about 400 years old. And I just wonder, what do we know about lifespan? Like what causes, some animals to, like my fish in the fish facility, they'll live for maybe 2 or 3 years, but then we have these other species that have been dated, although not proven, but it's thought that they're like 400 years old. Mm-hmm. Do we know anything about that?
Not much, I would say. About the sharks, I read that article too. These are deep sea sharks. And they're rarely sighted, and they can get their cartilaginous, but they in fact are believed to have lived for as long as 400 years, which is pretty astonishing.
They grow very slowly, though.
They grow very slowly, yes. But just take mammals, for example. I mean, mammals, I think, are a really good example to show you how little we really understand how these processes are being modulated by developmental biology, by evolution, etc. So nobody would argue that mammals descended from an individual species at some point in the past. Okay, so
well, some might argue.
Well,
but
there will
nobody
always
rational. Nobody rational.
Right. So there's no credible evidence to suggest that the mammalian group of or mammals are not what we call monophyletic, or single origin in time. Okay, and so if you now look at all of the extant mammals that we know of, okay, what you see is that they range in size from about 1.5 to 2.5 centimeters for the Madagascar shrew. It's a little mouse about this big, to of course the big sperm whales, which are bigger than this auditorium, right? It's the same genome, so to speak, right? And so that's about 1 million-fold difference in size and scale. And so that addresses the question of like rate of growth and longevity and so forth. But the longevity part is even, I would say, is equally interesting because the age span also ranges about 100-fold difference between all of these species. Now the question really is, how is it that, common ancestry from an original species way back in time could have actually evolved in such a way that it can actually occupy what we refer to as this genotypic, phenotypic landscape. And that we really don't understand. I mean, the dream would be that one of these days you can actually look at a genome sequence and know what it's coding. And I mean like what the animal looks like. Right now you can't. But it's all somehow codified there. And if this is a common origin and it still has all of this space where it can actually explore depending on the environmental demands or selective pressure to produce all these very specialized forms and longevity and size and scales, those are things that we don't understand. And I think that's where the next conceptual leap in our understanding of biology is probably going to come from.
Well, I personally would not want to live to 200 years if I had to grow a lot bigger than, But I'm going to now please invite people up to the microphone. There must be some questions from the audience. Anyone, please feel free.
Here
we go. Here's our first one, the icebreaker.
Great talk, thank you. The question about integrin, do you think we could inject it into a muscle of elderly people or people who have a stroke, and then do you think it can?
Well, that's what we proposed. Presumably it's possible, but there are other ramifications because integrin does many other things. In terms of stroke, that's actually a very interesting question. I think you're referring to people who actually in the hospital bed,
yes.
And they lose all the muscle. Actually, that's a very interesting question. There are some people study it, so this is one of the phenomena many people know. You don't have to have a stroke. As long as you are in the hospital under anesthesia, even just one day, you actually lose a lot of muscle.
Yes.
And we actually do not understand.
Disuse atrophy, right?
Yeah, so it is a very fascinating phenomenon. Yeah.
Thanks.
Anyone else have a pressing question? Yes, here's another.
Hi. You had shared a crystal structure of, I'm assuming, a protein.
Yes,
Would
your
be
assumption is correct.
classified
yeah,
as protein. Have you tried to see anything about the active site or changing just the smaller sections within that protein to figure out if it
Yeah,
does the same function?
Yeah, so I have not. I mean, that crystal structure was not generated by my lab. Crystal structure was generated by a collaboration between a group working in C. elegans and a group working in mammals. But your point is well taken, is that out of all of that beautiful structure you saw there, only some parts of it, okay, are going to perform the catalytic activity. And the rest are there for, its own reasons, right? But this particular, the reason why I show this particular crystal structure is because this actually came about from one particular genetic experiment that was done in C. elegans in the nematode, where beta-catenin, the first beta-catenin that was discovered, was actually doing the opposite of what beta-catenin in mammals was doing. Instead of activating, it was a repressor. And people thought that these worms were weird because, the beta-catenin is acting strange and so forth. But then later it was found that there was another molecule called cis1 that actually looked the phenotype looked just like it was an activator, like beta-catenin should be. But when you compare the sequences, they were so dissimilar from each other that they didn't think that it was the same molecule. But the geneticist who was in charge of this work, persisted. And she said, well, no, we're going to crystallize this. And so when they crystallized this molecule, what they found out is that when you superpose the cis1 protein from C. elegans with the mammalian protein on top of each other, they're almost identical. So even though the sequence was different, they're almost identical. And where they were most identical were these catalytic sites. So my suspicion is that there's a great deal of effort being invested in trying to understand what those catalytic sites may or may not be doing, such that you can figure out what kinds of things that protein may be doing in cell 1 versus cell 2, because those contexts will change. But I am not doing any of that work. But I'm leaving it to my colleagues to sort that out. And then I can use their slides.
So I can add on to that. So in case you wonder whether it does anything in muscle regeneration by either overexpressing or taking it out, absolutely nothing. So biology is very, very interesting.
Yes. Yes.
Right.
Yes.
Thank you.
No, thank you.
But there are other organisms like Hydra where it does similar functions, making more head, like affecting the head
Yes.
versus the.
Yes,
Yeah.
that's right.
Anybody else? Oh, come on, let's have a biologist come up here. We have a few. We have a few ringers in the audience.
Layperson. Yeah.
Okay.
So, okay, well, sort of fairly layperson. So I hear myostatin is, you're right, like super fascinating, and I encounter it when I'm reading about mechanisms that stop growth. So our liver knows to grow this big and only this big when it regenerates, and all these things know to grow this big but only so big. And myostatin has been proposed, and I really don't know the evidence, it's one of these things I encounter and then never follow up on, that it's actually a circulating
Yes.
factor.
Yes, it's a systemic factor. Yes, you're absolutely right. One of the most fascinating things about regeneration is what I said, proportional regeneration.
Yeah.
You cut off the liver, they will regenerate exactly the right size, more or less. So why don't they just grow bigger?
Like fuse, I think it's axolotls, and they only let one liver, but the two are fused, and then the one liver and the one axolotl gets twice as big.
Right. So there's a total size control.
Yeah.
So if you think myostatin is fascinating, I can tell you that Sagen actually did extra experiment to remove follistatin, and the muscle is even bigger. So there are even you can get rid of it. There are increase,
But if it's
not a
circulating,
limit.
like what's the
What's
like
the
what
limit?
stimulates it? Like what's the
Oh, what's
Receptors.
stimulated?
Well, maybe receptors.
Stimulated, actually, that's what
yeah,
tumor
I don't know the
does.
receptors. Is it like hormones or
What do you mean?
like
What
does
stimulates
it come from muscle? Does it come from muscle?
Actually, many tissue express it.
Oh, wow. Okay.
In fact, again, it's a mutant that give you big muscle, so you actually don't want to have it.
Yeah.
So Seijun always joke about his must be, and if anyone knows Seijun, he's skinnier than I am. He always joke about he's a myostatin overexpressor.
Yeah.
And the idea is presumably only when you inhibit it, you'll get bigger muscle.
So are you telling us you could make a double mutant and have an even bigger bull
Yes.
than the bull you showed us?
Yeah, those are heads.
Presumably.
Okay,
yes.
well that's a
And
scare.
by the way, since you mentioned the bull, I was told Belgian blue tastes just as good as regulum.
Not to a matador. and yeah, go to the microphone, please, my director.
So is it possible to actually inhibit these things, develop inhibitors to these things so you can actually use that to treat patients? So
And
the
that's-
myostatin.
Yeah, that's
what
the
they're-
activin receptor 2BR4.
So that's being
Actually,
showing
I
promise.
know at least 5 companies work on that. There are many, many use other than cancer cachexia. One of the main proposal, I believe, from Novartis is to help. What is that? What is that? What is old people when we fall to the floor? What is that?
Broken hips?
Yeah, so it's actually to prevent broken hips because typically the muscle here are the easiest one to shrink when you are growing older. That's why older people tend to have broken hips. So Novartis is trying to develop the inhibitor to strengthen the hip muscle to prevent it.
Here's another question.
Wonderful.
On that note, have long-term studies been done to see, you showed impressive results with muscle regeneration, but could things go wrong beyond
Very good
that?
question. And those are, so I know the myostatin one, it is still in clinical trial in the safety phase. So that's one. We have not done any of those, so we don't know for our situation. Or what I can say, those are 1 month after. That's for mouse, that's reasonably long. In human, we don't know. It's a great question. How long does it last? How many times do I need to reinject? I understood for Amgen, the argument is they did do aged people as a control, and I'm pretty sure Seijun told me it lasts for at least a month. Like to have muscle gain, you only need to inject once, and it will last for a month.
But could it mechanistically cause other things like a cancer?
We don't know. These are all need to have long-term studies.
It must be something that's going to be pretty popular with weightlifters.
Uh-huh.
Bodybuilders.
But I think I think if you take mouse model as a sort of predictor, myostatin mutant mice do not develop more tumor. So presumably by inhibiting it shouldn't cause that.
Do they live as long? Does it affect lifespan?
Actually, they live slightly longer, and their heart is healthier, and they have less fat. So that by definition, they should be good, and we still don't know why it's an inhibition. Why is that? Why do we have the gene? Right? If it's really good for you. Or is it really bad for you? Sorry.
Anybody else? Okay, well, I'll turn it over now to Yixin, and thank you for your attention.
Great discussion, and thanks for coming again. There will be refreshments out there, and we can continue to mingle. I think this can continue until 8:30. By the way, I just really want to say one thing. You are in this beautiful auditorium called Rose Auditorium, which is made possible by Deborah Rose, who's one of our trustees, and who we are very grateful, who came here to support our first public lecture. Deborah.
Sarah Lee. Nice talk. Very nice.
Okay. let's all go out there to continue our mingling and interactions.
Okay.